Okay, DNA contains the instructions for proteins. Those proteins control our functions and thus our traits. Now let's really dig into this. One of the things that we need to talk about in order to really make this make sense is nucleic acids. Nucleic acids are one of the categories of major biomolecules. The other ones that we talk about, proteins, carbohydrates, lipids. Nucleic acids are so named because they are long polymers that have an acidic character to them and are generally found in the nucleus. So that nucleic refers to being found in the nucleus, acid is about their chemical characteristic. There are two types of nucleic acid: deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. There's a lot of interesting chemistry, they're subtly different. We're not going to get into the whys and wherefores of which one is, different in what way and how they're used. Just know, DNA and RNA are very, very closely related, very similar in a lot of ways, but they are different and they do get used in different ways. The function of these molecules is information. They store information and they transfer information. Generally speaking, DNA, deoxyribonucleic acid, this is our long-term information storage molecule. This is what your genes are made out of. RNA, ribonucleic acid, this is your short-term information molecule. This is what we transfer from one place to another using. What we're going to talk about is DNA structure and DNA replication. But before we get there, I just want to get us kind of all on the same page using a little bit of some metaphors. We're going to talk about DNA replication, where you use DNA to make a copy of more DNA. And we're going to talk about the processes of transcription and translation, where you use that DNA to make an RNA copy, which then tells the ribosome how to make a protein. We talk about these processes around the same time because they involve a lot of the same chemistry, but they're distinct and they have very different purposes. Often what we talk about is a cooking metaphor, or rather a cookbook metaphor. The cookbook is the genes. This is, for me, my beautiful copy will save the joy of cooking. Big, heavy bound book. DNA replication is like if I had a way at home to take that copy of the joy of cooking and make a new copy of the joy of cooking. Not a big stack of ugly Xeroxed pages, but actually with the cover and the embossing and the nice sewn binding and all of that jazz. DNA replication is about making copies of the long-term information storage of that beautiful, heavy, well-bound cookbook. Transcription and translation. Well, transcription is what, I don't know about all of you. When I cook, it can be a bit of a nightmare. I try to be a neat cook, but I'm enough of a klutz that there have been some truly catastrophic incidents with pasta sauce and muffins and onion rings. And anyway, I generally have learned that it's not a good idea for me to have my nice pretty cookbook sitting right next to me as I'm cooking, because there's a good chance that there's going to be splatter damage or possibly fire extinguisher damage. So instead, I'm going to jot down a copy. Maybe I'll run off a Xerox while I'm at work. Maybe I'll just scribble the recipe down on a piece of paper. That's what transcription is. You don't want to risk your beautiful recipe book that's all bound and has tons of recipes in it just because you're making pasta sauce. You're going to scribble down the recipe with all the information in it on a random piece of paper that if it gets splattered in pasta sauce or accidentally catches fire or whatever, It's not a big deal. It was just a random piece of paper. So transcription is scribbling down the messy copy, looking at the original recipe book. Translation is the process of actually making the pasta, and hopefully this time not setting anything on fire or having giant pasta sauce splashes anywhere. So these are metaphors that I'm going to use a lot when I'm talking about this because, again, cell biology, everything is so small. We end up using a lot of sometimes slightly tortured metaphors in order to try and make it easier to grasp, something we can think about more clearly. All right, DNA and its chemical structure. So, nucleic acids, both DNA and RNA, are made of chemical building blocks called bases. Each base consists of a phosphate, a sugar, and then a nitrogenous base, all attached together. The way that these are hooked up is that we've got a backbone of alternating phosphates and sugars, each sugar having one base attached to it. If we're talking about DNA, the four bases that get used are A, adenine, T, thymine, C, cytosine, and G, guanine. So we've got the sugar phosphate backbone with these bases sort of sticking out from them. One of the things that's really important about DNA and RNA, about the nucleic acids, is that the bases pair very specifically. An A, an adenine, will always pair with a T, and a T will always pair with an A. There's no flexibility, there's no, oh, well, maybe this, maybe that. No. They base pair A with T, period. Cytosine pairs with guanine, guanine pairs with cytosine, CG pairing all the time. This means that even if we didn't have the sequence for that right strand on this particular one, I would still be able to tell you, well, if the left strand is A, G, T, A, C, G, then that means by definition, without me having to see that other strand, it is going to be from top to bottom T, C, A, T, G, C. Always. DNA and RNA, if it is double-stranded, is going to be in this double helix shape. Just fundamentally, by the way that the bases are shaped in space, when you get the long polymer, you have your sugar phosphate backbone. So just note, along here we've got all of our sugars, and they're connected by our phosphates, with the bases being like the rungs of the ladder and the sugar phosphate backbone being like the long straight parts, but it twists. Inside of your cells, rather, your chromosomes are absolutely enormous. When they're packed up for mitosis, like they were in that karyotype, they're fairly compact, but getting there takes a lot of work, because in one single cell of yours, something that is too small for us to really be able to see clearly, there's between 1 and 2 meters of DNA. That's sort of like taking a smallish house and sticking 20 or 30 kilometers of yarn inside of it. If it's just giant gnarly piles, nobody's going to be happy. But if you have it nicely twisted and spooled up and carefully packaged, it isn't that big of a deal. And so, in this case, DNA gets wrapped around these little proteins that are called histones, and then that creates a coil which itself can be coiled, which itself can be looped onto a framework, and through a lot of packaging you can get to a nice compact chromosome. So the process of replication means using DNA as a template to make more DNA molecules. That's this chunk. Again, this is as if I could take my copy of The Joy of Cooking, and obviously with a lot of work and skills that I personally don't have, probably some equipment I certainly don't own at home, but if, let's say I took up bookbinding as a hobby, I took that copy of The Joy of Cooking and I very carefully made exact copies of every single page. I glued them, I bound them, I put the cover on, I did all the embossing, and at the end I now have 2 perfect copies of The Joy of Cooking. When do you do this? Why do you do this? The cell does this when it reproduces, when you're going to go from having 1 cell to 2, because those 2 cells have to have all the same genetic material as their parent, otherwise they're going to be different. Otherwise something gets really screwed up. You're not reproducing yourself, you're creating some crazy who the heck knows what it is thing. So, you have to be able to very carefully and accurately, precisely, make copies of every single part of the DNA. Because of this characteristic of DNA, where every A is paired with a T, every T is paired with an A, every C is paired with a G, every G is paired with a C. Because of that, it is possible to split the DNA molecule open, as is happening here. So we've got it opening up, and a new strand can be manufactured. And a new strand can be manufactured using that parental strand as a template. So over here with our red and blue molecules, the idea is that this red strand has split open, enzymes have come in, brought in nucleic acid monomers, brought in nucleotides, and connected them up so that they've now made this blue strand, resulting in 2 complete and identical DNA molecules. Now, as with so many things that we've talked about, it's not quite that straightforward. There are a ton of different enzymes that have to get involved in this. We've got leading strands, lagging strands, issues of how the nucleic acid chemistry works, etc. But just know, this is a process that happens. You get 2 identical copies of every single chromosome being made during this. So now not only do you have 1 chromosome from mom and 1 chromosome from dad, the chromosome from mom itself has been copied. The chromosome from dad itself has been copied. And all of this happens in a very regulated fashion. All these enzymes, the topoisomerase, the helicase, the DNA polymerase that are pictured in the lower left, these are only going to be around and active when it's time for the DNA to replicate, when it's time to make a new cell. The rest of the time, they're either gone or they're turned off, because you don't want to do this unless it's time to do it.